Hi. So in previous session we have learnt about various PRR, and among those PRR we have extensively studied toll-like receptor, and we have also studied Rigide-like receptor RLR. Okay. So these receptors are playing very important role in various compartment of the cell. You have seen that toll-like receptors are present on cell membrane, okay, outside the cell membrane, and it is also localized in the endosome, okay. And you have also studied that there are sensors which we call it as a Rigide-like receptor. They are present in cytoplasm, okay. And so now the whole cell is fully guarded. If you see carefully, fully guarded against various invader or microbial pathogen or its component. Okay. Today we will discuss another very big family of pattern recognition receptor, and these pattern recognition receptor we call it as a nod-like receptor. Okay. We will discuss about nod-like receptor. We will discuss about the various microbial pathogens sensed by various member of nod-like receptor, and we will also discuss the signaling pathway which is activated by these NLR members. Okay. So let us begin with nod-like receptor, and nod-like receptor is or NLRs are first identified in plants. It's very interesting, right? It's first identified in plant, and they play a very important role against microbial and parasite infection in plants. Okay, and these proteins are named as R genes. Okay. So R genes are basically R is simply resistance gene, okay, and this resistance gene is very important in defense in plants. So this is far more interesting. You have seen that for example, the phagocytosis is originally discovered in the starfish larvae, and you have seen the toll protein was originally discovered in Drosophila, the fruit fly. Here you are seeing that this nod-like receptors, they are present, or their members are also present in plants, okay, and they play a crucial role in defense. Okay. So the homologue of this nod-like receptors are present in vertebrates. It is present in most of plants. It is present in vertebrates, and it is present in very primitive or phylogenetically primitive organisms such as sea urchin, zebrafish. Okay. And in these animals the about 203 members are present. So you can understand that these proteins are kind of very crucial for defense in these primitive organism, okay, like zebrafish, sea urchin, or so and so, and they play a very important role in various vertebrates. It is present in almost all vertebrates. Okay. So you can understand how primitive this this gene is, okay, and this is highly conserved. The whole NLR family members are highly conserved in sea urchin and vertebrates, mice, human. Okay. So all these members, the nod-like receptor family members, they are playing very important role in host defense. And in human, about 22 members are there, 22 NLR members are there, and in mice about 33 members are there. I will show you in next slide. And these members are having a, if you categorize these protein, then these proteins have a two distinct functional feature. Okay, one is that they directly sense, and then it induces a activation of or NF kappa B or MAP kinases in order to produce pro-inflammatory cytokines. Another group of proteins, they make a multi-protein complex, okay, and they play important role in production of one very important family of cytokine, which we call it as a IL-1 family cytokine, and they are also playing a various role or important role in one or other types of cell death. Okay, that includes apoptosis, pyroptosis, so and so. Okay, so these are the functional type of node-like receptor. There are two major categories. Okay, so we'll look at all this thing. If you see this slide, here you can see that in human there are various node-like receptor members are there, and in mouse their homologs are there, and their subtypes are also there. Okay, in mouse, and in and you can see the domains in these protein. So I will begin with CIITA. It's a class 2 trans activator protein, and this class 2 activator protein has a card domain. Okay, you remember the card, caspase recruitment domain or caspase recruitment and activation domain. Okay, I have discussed in previous session in RLRs. Okay, so class 2 trans activator protein, they have a card domain, they have activation domain and NOD domain. Okay, so NOD is basically nucleotide binding oligomerization domain. Okay, so basically it binds with nucleotide and that induces the oligomerization. Okay, and there is a leucine rich repeat. You probably remember leucine rich repeat is also present in toll-like receptors. Okay, and over there it is present in ectodomain. Okay, which basically play an important role in sensing the PAMPs, various PAMPs. Okay, so this is a class 2 trans activation protein. Another is NAP, NAIP, and they have a BIR domain. Its BIR stand for baculovirus inhibitor repeat. Okay, and there is a NOD domain and leucine rich repeat domain. So NAP is also making some or other kind of multi-protein complex, and then it is playing important role in production of or induction of mature form of IL-1 family cytokine. Another is NOD1 and NOD2. So NOD1 and NOD2 is basically present in cytoplasm, and they sense some key motifs of peptidoglycan. Okay, and basically it activates NF kappa B and MAP kinases. Okay, so you can see that NOD1 and NOD2 both has a, NOD1 has a one card domain and NOD domain and leucine rich repeat domain. On another hand, NOD2 has a two card domain, NOD domain and leucine rich repeat domain. Okay, so here if you notice in these protein NOD1, NOD2, NLRC3, NLRC4, NLRC5. So all these proteins, they have a card domain, and this is based on domain classification. these proteins are basically grouped together and they are called it as a NLRC, card domain containing NLRs. Okay. NLRC3, if you see, this has a one domain which is people are kind of not clear. This is either card domain or there is a pyrene domain. Okay, and they have a NOT domain and leucine rich repeat domain. Similarly, in case of NLRC 4 and 5, there is a card domain, NOT domain and leucine rich repeat domain. So, here just I want to give a note. So, there are so many members, but all members are not very well characterized, but few members are extensively characterized. So, I will tell you in upcoming slides. Another class, if you see, this has a NLRP 1, 2, 3, 4. Okay, and if you see here carefully, they have a pyrene domain instead of card domain. So, they have a pyrene domain, NOT domain, leucine rich repeat domain, and in case of NLRP 1, there is a both pyrene domain and card domain is present. Okay. So, NLRP 2 and NLRP 3, they have only pyrene domain. In addition, NLRP 4 also has only pyrene domain, NOT domain and leucine rich repeat domain. So, among all these NLRPs, NLRP 3 is very well characterized. It is extensively, or I think it is too much characterized. It's everything we know about the NLRP 3. Okay. It's the mutation associated with that protein, the type of ligand on by which this NLRP get activated, and how this NLRP get activated, and what are the other components, other proteins which interact with NLRP3. Everything is quite well characterized. Okay. Here there is a another member, NLRP5. This also has a pyrene domain. Here you can see that this has a pyrene domain, NOT domain and leucine rich repeat domain. And rest of another NLRPs are also there, NLRP 6, 7, 8, 9, 10, 11, 12, 13 and 14. So all these NLRPs has a characteristic pyrene domain, NOT domain and leucine rich repeat domain, except if you see little carefully, NLRP 10, this does not have a leucine rich repeat domain. So, and the last member is NLRX1, and this has a NOT domain and leucine rich repeat domain, and there is some region which is not very well defined, which will fall either card domain or pyrene domain. It's not very well characterized. So, NLRX1 is like that. Okay. So, now I will show you what are the ligands these protein senses. I will not talk about all proteins. As I told you, all of these proteins are not very well characterized. I will talk few of these protein which is quite well characterized. So, here you can see there is a NOD1 and NOD2. This has a leucine rich repeat domain and nucleotide binding domain or NOD domain or card domain. Okay. So, NOD1 basically, since IEDAP, I will explain you IEDAP more detail in next slide. So, IEDAP is basically derived, it is a component of peptidoglycan. IDAP is okay. I will tell you precisely what is molecularly it is, and this IDAP is derived from Listeria monocytogen. You can understand this is a bacteria, Chlamydophila pneumoniae, Asteraceae coli, Shigella flexneri, Campylobacter jejuni, Chlamydia trichomatis, Pseudomonas aeruginosa, Haemophilus influenza, and Helicobacter pylori. So these are the microbes which is sensed by NOD1, and what it is sensing? It is sensing IE-DAP. IE-DAP is a one motif or one region in peptidoglycan. On another hand, NOD2, which has a 2-car domain, they sense the MDP. Again, I will tell more about the MDP. Basically, it's a muryl dipeptide. Probably you may aware about that, which is it senses MDP. Mycobacterium tuberculosis. It also senses again Listeria monocytogen, which is also sensed by NOD1. Streptococcus pneumoniae. Okay. So once these NOD1 and NOD2 senses these particularly these pure ligand, one is IE-DAP and MDP in the cytoplasm, it induces the production of inflammatory cytokine through activation of NF-kappa B and MAP kinases. Okay. So this is one functional category. Okay. Another functional category is this is basically NLRP1, NLRP3 and NAP5. So these protein basically senses the ligand, and then it will make a multi-protein complex. Okay. And this multi-protein complex basically induces a very important cytokine, which is or family of cytokine, which is we call it as a IL-1 family cytokine. Okay. And this IL-1 family cytokine basically consists of several cytokines, but key cytokines are IL-1 beta, IL-18 and IL-33. Initially, these cytokines are synthesized in inactive form, and once these protein get activated, this makes a multi-protein complex, and then they will induce this mature form of IL-1 beta. I will tell you the signaling in subsequent slide. So here you can see that NLRP1 basically senses the MDP and lethal toxin of Bacillus anthracis. Okay. So Bacillus anthracis has three major protein which is making the exotoxins. Okay. So there are, probably you are aware that there are two major kind of toxins produced by the bacteria. One is endotoxin. Endotoxin is like LPS. Exotoxin, they are toxin which is synthesized in the bacteria, and then it is secreted out. Okay. So Bacillus anthracis makes this lethal toxin along with two more proteins which we call it as a protective antigen, and there is a edema factor. Okay. So this protective antigen basically helps in transportation of lethal factor and edema factor inside the cell in order to kill the cell, okay, target cells. So I will not go in more detail about the Bacillus anthracis, but this is just for your information. It's a special note you can remember. Okay. And this basically induces caspase-1 dependent cell death. Okay. So NLRP1 is basically involved in cell death. So I have told you in beginning of this session that functionally they have two major category. One is activating NF-kappa B and another is inducing IL-1 family, mature form of IL-1 family cytokine, and another category also induces cell death. Okay. So this basically this NLRP1 is inducing the cell death, and it is caspase-1 dependent. Another is NLRP3. As I told you, this is very well characterized NLRP family member. Okay. We also call it as a NALP3. Okay. And CIAS1 cryopyrene. Okay, so this cryopyrene is or this protein, they sense plethora of ligand. It's a huge list. I am putting very few, not all. Okay, and they can sense, if I see the literature, they can sense all kind of ligand, living, non-living, toxins, and so on and so forth. Our own antigens or our own molecules they can sense, and they will induce production of IL-1 family cytokine. Here I am naming few of the ligand. Here you can see, this can see the MDP, they can sense microbial RNA, R837, this is a R837 and R848, these are the ligand which activates TLR also. They can also sense various TLR ligand, they can sense the ATP from the host also. Okay, they can sense various cytolytic toxin like niagricin, areolysin, gramicidin, and alpha toxin and metotoxin. Okay, these NLRPs can also get activated by some physical mean. Here you can see there is a ultraviolet radiation can activate this NLRP3, and this is also activated by some chemical compound which is produced in our body. Here the best example is MSU, monosodium urate. Okay, this monosodium urate is increased in the individual who has a tendency to have a gout, okay, or joint pain. Okay, so MSU is a one of factor which is responsible for the pathogenesis of gout, and another is CPPD. It's calcium pyrophosphate dihydrate. So this is also produced in our body. So basically monosodium urate, this is produced more in those individual who has some problem in nitrogen based molecules, and you know that amino acids are all nitrogen. Okay, so if there is a problem in metabolism, then that may result to the accumulation of monosodium urate, and that will be one of key cause for this gout. This can also NLRP3 can also activated by our vaccine adjuvant. As I told you, in human we use only alum as a vaccine adjuvant. Okay, so this can be activated by alum, this can be activated by silica. Silica, you know, the silica is the people who are working in cement factory or similar kind of situation where there is a generation of lot of silica dust. So those individual may develop some or other disease. Okay, so this silica can also activate NLRP3. Besides this, some chemical compound like picryl chloride, which we use it in our experiment. Okay, and beta amyloid protein. So all these things, these are very short list of NLRP3 ligand. So all these proteins basically activate the NLRP3 inflammasome. There is a new term here I am telling, inflammasome. Okay, so this NLRP3 makes an inflammasome, not NLRP3, other NLRPs also makes a inflammasome. So what is inflammasome? So inflammasome is nothing, it's a simple multi-protein complex, and the aim of this multi-protein complex is to activate the enzyme which is present in inactive form, and the enzyme is procaspase. Okay, it's a procaspase 1 Okay. In case of mice, it is procaspase 1 and 11. Okay. So procaspase 1 is enzyme which cleaves the pro IL-1 beta into active form, that is IL-1 beta. So try to understand here one simple philosophy that this IL-1 family cytokines, they are quite potent. Okay, it's a small doses is quite sufficient in order to develop or induce significant biological effects. For example, IL-1 beta, they can induce fever. Okay, they can, if it is produced too much, they can induce fever, high fever, or they can induce the hypotension, and that is a quite big physiological effect. Okay. They can also make condition like a shock condition. IL-1 beta is also one of the key factor. Okay. So they make a multi-protein complex, and this multi-protein complex, the aim is to activate the caspase which is present in inactive form, that is procaspase, and once this procaspase is activated, then caspase will generate, and this caspase will make the active IL-1 beta. So this inflammasome are various kind. I will show you in subsequent slides. Another is NEP5, and this NEP5 has a this senses this Legionella pneumophila, and they also control the replication of bacteria independent of caspase 1. Here the caspase 1 is not involved. There is another inflammasome which senses the flagellin protein of Salmonella typhimurium, Legionella pneumophilia, and Pseudomonas aeruginosa. Okay, and this inflammasome also induces caspase 1, IL-1 beta, and IL-18 production. Okay, so you can see there are two distinct categories. One is inducing or activating the NF-kappa B and MAP kinases, and another is basically inducing inflammasome mediated cell death or production of IL-1 family cytokine. Okay. So now I will explain you what is this. You have seen previously there is a ligand for the NOD1 and NOD2. So here you are very well aware about the structure of gram-negative bacteria. I am just showing you for your quick understanding. So there is a outer layer which is consist of LPS, okay, outer membrane, which contains LPS, and there are other things like porine and lipopeptide, and there is a peptidoglycan. And then there is a membrane, okay, plasma membrane in case of gram-negative bacteria. And in case of gram-positive bacteria, there is a very thick peptidoglycan layer, okay, which makes a cell wall. And there is some projections of polysaccharide, and of course below the peptidoglycan there will be a cell membrane. So this is a more detailed structure of peptidoglycan. This is basically consist of or it is a polymer of NAG, N-acetyl glucosamine. Okay, if you remember your previous classes, or you probably studied in microbiology. Okay, so NAG is an N-acetyl glucosamine, and this is linked by N-acetyl muramic acid, and this linkage is basically beta 1,4 glycosidic linkage. Okay, so this N-acetyl glucosamine and N-acetyl muramic acid, which is linked by glycosidic linkage, is basically there is another molecules which is making a kind of cross linkage between two these polymer chain, okay, polymer chain of NAG and NAM. Okay, and this is basically linked by L-alanine, D-glutamic acid, mesodiaminopimelic acid, here you can see, and there is a D-alanine. Okay, so this is a molecule where you can see both D and L amino acid, that is L-alanine and D-alanine, both are present, and this tripeptide chain is again linked with here you can see this is having a D-alanine, okay, and meso diaminopimelic acid, and then there is a L-alanine. Okay, so these two chains of this NAG and NAM is linked like that. Okay, and here you can see this MDP is a muryl dipeptide. So this muryl dipeptide is basically this component, which is consist of NAM and acetylmuramic acid, L-alanine and D-glutamic acid. So this component is sensed by NOD2. Okay, and there is a another component. If you see below, then you can see that there is a diaminopimelic acid, okay, and D-glutamic acid. Okay, so this D gamma glutamyl meso diaminopimelic acid is a IEDAP, and this is a ligand for NOD1. Okay. Let's look at how this NOD1 and NOD2 activate the signaling. Okay, so this is a very simple schematic. Try to understand. I am again saying it is a very simple, but it is far more complex. Okay, for your easy understanding, I made it very simple. So this NOD1 and NOD2, upon sensing this ligand, they make a oligomerization. There will be a conformational change, and this conformational change will make it oligomer, and then this will recruit another card containing protein, which we call it as a cardic. Okay, and this cardic is a key adapter kind of molecule, and this will recruit another card containing protein known as card9, and then this can activate MAP kinases, and cardic directly can activate the NF kappa B, and you know that how NF kappa B get activated. It is activated via IKK complex, which phosphorylate I kappa B, and this phosphorylated I kappa B undergo degradation, proteasomal mediated degradation, and then this free NF kappa B will be translocated into the nucleus, and then there will be a production of inflammatory cytokine. So this is the way by which NOD1 and NOD2 activates NF kappa B and MAP kinases. Okay. Now I will talk about the activation of inflammasome. Here you can see there are various kind of inflammasome. There is a NLRP3 or NALP3 inflammasome. There is a NLRC4, NLRC, NAP5 and NLRP1 inflammasomes are there. And now I will tell the composition of inflammasome. So basically this inflammasome is basically consists of the NLR family member. In case of NLRP3, it is a NLRP3, and there is a one molecule which is also present, which we call it as a ASC. Okay, so ASC, if you see carefully, this ASC has a both pyrene domain as well as card domain. Okay, and it will interact with procaspase. Okay, this procaspase is basically having a card domain, okay, and caspase domain. So here you can see this homotypic interaction is playing very important role in making this multi-protein complex. Okay, and then this complex, this multi-protein complex, get activated by the ligand, and then it will induce the active form of IL-1 family cytokine. Okay, here you need to note one very important thing. In order to activate this inflammasome, you need a two signaling. One is that any signaling which will increase the amount of caspase 1 as well as pro IL-1 family cytokine. which is driven by NF kappa B. Here you can see there for your convenience I put it TLR. Okay. So there will be a TLR mediated NF kappa B activation. Okay. This NF kappa B activation will result to the more amount of protein in the cell, more amount of procaspase, IL-1 and ASC. Okay. This is signal 1, and signal 2 basically make the oligomerization of this NLRP family members. Okay. And these two signaling work together, and then through this inflammasome multiprotein complex there will be a processing of procaspase, and this procaspase will first make a caspase, active form of caspase, and this active form of caspase will cleave the pro IL-1 family cytokine, and then there will be a generation of IL-1 family, active IL-1 family cytokine, which is secretory in nature. Before cleavage it is not secretory. Once it is cleaved, then this will be a secretory. Okay. So this is a basic concept of inflammasome. There are various kind of inflammasome like NLRP1 here you can see, which is activated by lethal toxin, MDP, ATP, and here NLRP3 inflammasome. There is a quite big list, and this list is further very long. Okay. It is activated by PAMPs as well as DAMPs. PAMP is a pathogen associated molecular pattern which is originated from the pathogen, and DAMPs are basically our own molecule, damage associated molecular pattern. Okay. So this can be activated by our own molecule also. So this is about NLRP3 inflammasome. There is another inflammasome which we call it as a NAPE or NLRC4, and this is also making a multi-protein complex, and there is a cytoplasmic DNA sensor, which I will talk more in when I will take up sensing of DNA most likely in next session. Okay. So this also make a inflammasome which we call it as a AIM2 inflammasome. So now I gave you the overview of NLR family pattern recognition receptor, how many members, what are the ligands, what kind of signaling, everything you probably understood. So in next session I will talk about the disease associated with NLR family members. Okay. And with this I will stop here, and in next session we will discuss about the NLR and disease. Thank you.